Alright, this video is going to talk about our neuroglia. These are those supporting cells that help to take care of the neurons because they've lost the ability to function completely on their own. Sometimes you'll hear neuroglia referred to as glial cells. Now, we have more variety of glial cells in the central nervous system, but we do have a couple of types that are present in the peripheral nervous system. We have 4 types of glial cells in the central nervous system that we want to talk about. The first one is the astrocyte. The astrocytes are the largest and the most numerous type of glial cells found within the central nervous system. Their job is to create the blood-brain barrier. Basically what they do is they line the capillaries that exist in the brain, and they prevent many of the typical compounds that we associate coming out of the bloodstream from doing so in the area of the brain. That is because a lot of these could actually interfere with the functioning of our neurons. In addition to this very important function, they also create a framework for central nervous system neurons to arrange themselves on. Now, just as a side note, because I just realized I forgot, the blood-brain barrier is one of the reasons why treating diseases and disorders of the brain can be very, very difficult. Because of that blood-brain barrier preventing a lot of chemicals from coming through, it also limits the number of drugs that we are able to effectively use within brain tissue. So, for instance, let's say we're talking about cancer, just because that occurs pretty much anywhere in the body. The number of drugs that you could use as chemotherapy for cancer are actually, we've got a pretty wide range of them. But of that wide range of drugs, there's only a select few that you can use for certain types of brain cancer. And that is simply because there's only certain types that can get past that blood-brain barrier. Now, this framework. When I think of this framework, I almost think of like a lattice on the side of a house where you would have maybe ivy or climbing roses or something like that. The lattice work would be like the job of the astrocytes to provide that actual physical support or the framework so that the neurons are actually arranged properly. Lastly, they also help to repair damaged tissue. Now, this ability to repair is obviously very, very limited, but they can perform some minor repairs on the neurons themselves. The second type of cell is an oligodendrocyte. Oligodendrocytes are actually very important within our central nervous system, and we think that certain disorders of the central nervous system are as a result of problems with the oligodendrocytes, for instance, Alzheimer's disease. What oligodendrocytes do is they send out cytoplasmic extensions of their cells. So it's kind of like your arms reaching out, and those ends of those extensions actually wrap around the axons of the neurons within the central nervous system. This creates an insulation sheath that we call myelin. Now, each cell extension only does a short segment, but each oligodendrocyte can reach out and wrap short segments on several different axons. So if I look at an axon, it is not going to be covered by a single oligodendrocyte. And that's actually a good thing, because if something were to happen to a particular oligodendrocyte, then the entire myelination on that axon would be lost. And the effect would be very similar to a short circuit. That's kind of what that insulated myelin sheath is like. If you think of wiring in your house, it's very much like that colored rubber type insulation that goes over wires, and it prevents them from short circuiting. When that insulation is gone, though, that doesn't happen, and the circuit or that wiring area short circuits and it doesn't function. And that can happen with neurons as well. Now, a covered axon, where it is covered, is referred to as myelinated. Now, there are some small gaps in the coverage, and these are important, and we'll talk about why when we talk about impulses, and those are referred to as the nodes, or the nodes of Ranvier. The area of the axon itself that is covered is referred to as the internode, and you'll see that on several diagrams within your textbook. Now, why is myelin so important besides this insulation idea that we talked about? Well, not only does myelin insulate, but it also increases how fast an impulse travels. And again, you'll see why when we talk about impulses. In addition, it makes the axon appear white. And as you may know, we have two colors essentially associated with our brain tissue and our spinal cord tissue, gray and white matter. So the white matter are simply those myelinated axons. It's not that they functionally necessarily are different, other than how axons are different in function than other parts of the neuron, but it's not like they're doing one job and the gray matter is doing another. If you recall, we actually talked about what the gray matter is when we discussed the neuron structure itself. The third type of glial cell is the microglia. These are the smallest and the rarest. These are the leftover remnants, if you want to think of it, of phagocytic cells that used to be white blood cells in the central nervous system. If you think back to that blood-brain barrier, that blood-brain barrier prevents white blood cells from exiting the blood vessels, even in the area of the brain. And that's a problem because that would leave our brains and spinal cord areas completely defenseless. Well, what happens is that as we are developing as a fetus, at some point that blood-brain barrier kicks in and prevents that from happening, prevents that movement of the white blood cells. But before that blood-brain barrier is in place, white blood cells are free to move wherever they want. So what happens is, until that blood-brain barrier kicks in, the white blood cells actually do go and reside within the central nervous system. Once that blood-brain barrier starts, though, not only are the white blood cells that are in the blood prevented from coming into the brain, but the ones that are already in the brain and the spinal cord are prevented from leaving. So they stay, they differentiate from a typical white blood cell and keep that phagocytic role. They keep that immune and protection role that they would have in the regular bloodstream, but they look a lot different. They become much more highly specialized for functioning within the confines of the central nervous system. The last one are the ependymal cells. These lie in an area that we call the central canal of the spinal cord, as well as the chambers of the brain that we will discuss at a later time. When they are taken together, they create a lining that is called the ependyma, and this produces and helps to circulate our cerebrospinal fluid. Now, in looking at this diagram here of the different types of glial cells in the central nervous system, first orient yourself up here with where we are. This is actually a cross-section of your spinal cord, and you can actually see here you've got the darker tan, which is your gray matter, and then the lighter cream, which is the white. So we are actually zooming in on a piece that has some gray matter as well as some white matter. You can see within the gray matter we have the cell bodies of the neurons, and they're actually getting pushed up against by these astrocytes. You can see that astrocyte connections change, and so sometimes the leftover pieces are kind of left behind. In addition, we have these cells here, which are the oligodendrocytes, that have these extensions that go out and wrap around, forming layers around the axons, forming that insulation. So you have these areas of coverage, the internodes, and then the gaps are called the nodes. Back to the astrocyte here for a second, you can also see out this side how they have extensions that go and they completely line this capillary, secreting that chemical that generates that blood-brain barrier. You have the microglia here, and that's very small and very movable, and you can see why it has to be so tiny. It has to be able to zig and zag in and out of all of this web-like structure within the central nervous system. Last but not least are the ependymal cells. This is the central canal that's basically in the middle there of the spinal cord, and they line that area, secreting their cerebrospinal fluid, and as well as putting in the nutrients and hormones that the central nervous system needs to function correctly. So those are the four types of glial cells within the central nervous system. Now, the ones that are in the peripheral nervous system are a little bit different, and we only have two of them. The first one are the satellite cells. Their job is to surround and support the cell bodies in the peripheral nervous system. So, in some essence, they're actually acting almost like the astrocytes. But unlike with the astrocytes, there is no blood-brain barrier because we can't isolate the neurons from the surrounding tissues that they are embedded in. Otherwise, they can't interact. The second type of cell is the Schwann cell. This is actually named after a German scientist named Theodor Schwann, and these function very much like oligodendrocytes. They are there to cover the axons that lie outside of the central nervous system. The outer surface of the Schwann cell is called a neurolemma. Each Schwann cell, very much like the oligodendrocytes, only covers one segment of an axon. But because we have a bundling of function as well, okay, sorry about that, we had a little interruption there, but the Schwann cells can actually enclose several different axons. Now, this is within one Schwann cell, and they don't make the cytoplasmic extensions like the oligodendrocytes do. What happens is in the peripheral nervous system, if you think about your finger, you have both sensory receptors in there, and you also have motor neurons because you're able to move them. So a lot of the innervation in your peripheral nervous system is based on location. So what the Schwann cells will do is that they will actually bundle together sensory and motor neurons since they are coming to or from the same location. And you can actually see this here. This first one is a single Schwann cell that is just wrapped around a single portion of an axon. And that's kind of typically what you would think of like with an oligodendrocyte. But with this one, the cell is actually in the middle, and it's got about 10 different axons that are kind of pushed into it, almost like fruit embedded in jello, so to speak. So, for instance, these axons up here could be axons of sensory neurons, whereas these axons down here could be axons of motor neurons. But because they're in the same body location, they're bundled together using the Schwann cells, which actually saves space. Now, this myelin that they create as well is very important, not just in the central nervous system, but also in the peripheral nervous system. So we have a series of disorders that are called demyelination disorders. Basically, what happens with demyelination is we get progressive destruction of the myelin sheath on the axon. Now, there are some serious diseases that cause this, and we're going to highlight one of them, but demyelination can occur with something as simple as inflammation. That can actually cause the myelin cells to be destroyed, either the oligodendrocytes or the Schwann cells. You can get that from axon damage and also neural tissue scarring. And this can actually create a lot of problems. You can get things like sensation loss and loss of motor control, just depending on the specific neurons that are affected. One of the major diseases that is associated with demyelination is multiple sclerosis. And there have been several famous people who've had multiple sclerosis. The one that comes to mind immediately for me is Richard Pryor, who is a very famous comedian. And he battled with MS all of his life. Typically, when we see multiple sclerosis take place, it's going to affect the optic nerve as well as the brain and the spinal cord. One of the first ways that it comes to be noticeable in terms of symptoms is through speech problems, but it can also come to pass with vision problems. So those are usually kind of the gateway symptoms that will indicate that maybe that this could potentially be multiple sclerosis. You also can end up with balance and coordination issues because, again, that's your motor control taking place. Some other demyelination disorders include things like diphtheria, which is kind of rare here in the United States simply because we get a series of vaccines. The DPT, it's diphtheria, pertussis, and tetanus. It's a combination vaccine that you get multiple times, and it's been very, very effective at preventing diphtheria-related demyelination. The one that's actually probably more common, even though it's very rare in itself, is Guillain-Barré syndrome. Basically, you end up with a series of symptoms, and there's no one specific cause of Guillain-Barré, but whatever is going on with, however these various viruses or bacteria mutate, they start to affect the myelin, and you get this progressive destruction. The last one is heavy metal poisoning, and this would include lead poisoning, but I specifically worded it as heavy metal poisoning simply because there are other heavy metals out there besides lead that are a problem for us. Basically, this goes in and it actually destroys the myelin. So you can understand why this idea of lead poisoning can be very serious in young children whose nervous systems are still developing. By destroying the myelin, you're actually interfering with their nervous system, and this can have a very wide range of effects, from motor skill issues to learning issues to emotional issues. It just depends on where the destruction actually takes place. So now that we've talked about the neurons and the glial cells, let's take another quick look at some of the organization things of a nerve, just as a little bit of review. In the peripheral nervous system, the cell bodies, which compose the gray matter, are actually grouped together in little clusters that we call ganglia. The white matter is all of the axons that get bundled together in nerves, like with the Schwann cells. In the peripheral nervous system, we have spinal nerves that connect directly to the spinal cord, and cranial nerves that connect directly to the brain. The nerves within the central nervous system contain both sensory as well as motor neurons. In the central nervous system, cell bodies with similar functions are grouped together in areas called centers, and these would be like the processing centers of the various areas of the brain. The neural cortex is a very thick layer of gray matter that covers certain portions of the brain surface. We also can talk about a tract. And a tract are bundles of axons. They are white matter, and they share some common characteristics. So you have these tracts that run vertically, usually in the spinal cord. They start in the same place, they end in the same place, and their functioning is the same. Within the brain, we have pathways, and they link the centers of the brain with the body. So for instance, we have the visual center, the taste center, and they're all in different locations, but they have to be connected to the body in order for those to work. That example is, for instance, a sensory pathway where we deliver sensory information from our receptors to the processing center. We can also have a motor pathway, leaving from the central nervous system motor center to the skeletal muscle that they control. So that kind of wraps up our look at some of the cells and the organization of the central nervous system. And moving on from here, we're going to look at impulses, in other words, those action potentials that neurons are able to generate, and how they move, and how they're able to actually jump the gap at a location called the synapse.